The last lecture of the morning session is by Dr. Lilach Avitan. She was Australian superstar in STEM, and currently she's a superstar in computational neuroscience. Thank you very much for coming all the way to talk to us. Yes.
Okay. So I'm going to switch gears a little bit from the previous talks, and we'll talk about one of the greatest problem of the 21st century, I think. And that's, I'll start by calling it the amazing machine called the brain. So I'll start with just putting a few numbers here so we can understand the dimensionality and the complexity of the problem. So in the human brains, we have 10 to the power 11 neurons. There's about trillions of connections between these neurons. And in 1 millimeter cube of tissue, we have 4.1 kilometers of wires. Okay. And despite it being so massive, it only takes 2% of our body weight and demands only 20% of our energy. So that's quite an efficient machine. Okay, so what can this machine do? Well, this machine stores our memories, our sensations, our expectations, emotions, desires, plans for the futures, plans for tomorrow. So comes the question, how does the brain do it? We have so many units, they are connected in so many different ways. How can we have this coherent understanding of the world? And that's one of the major questions in neuroscience. How does the brain do it? Now, in order to understand how does the brain do it, we first need to understand what the language of the brain. So the brain speaks in electrical pulses, okay? Every neuron, when it wants to say something to its neighbor, it just sends zeros and ones. Tack, 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 and that's it, that's the code, right? That's the neural code. We have no understanding what this code is, but we do understand that the world is converted into this code, and this code is then propagated in different places throughout the brain, and then the animal or us are doing something or acting upon the world. All right, so not long ago, perhaps a few decades ago, we believed that single neurons hold some information. And in fact, there was a very famous paper in Nature that and headlines were that Jennifer Aniston strikes a nerve, okay? Because scientists found a neuron, and this is the zeros and one of this neuron, and this neuron was active only when the neuron saw Jennifer Aniston's photos, okay? So you could see Jennifer Aniston here and here and here and here. It's very hard to see because small pictures, but you could see that this is over many trials, this neuron is firing, okay, executing this or eliciting these electrical pulses. You can see the histogram of the response of this neuron. But if this neuron was shown a picture of Jennifer Aniston with her spouse, I guess, then this neuron didn't really like it. It didn't really also like any other celebrity, not Julia Roberts, any NBA players. No, this neuron really preferred Jennifer Aniston. It didn't really also prefer any famous architectural structures like the Eiffel Tower or the Opera House in Sydney. It just didn't, it wasn't really interested. It was interested in Jennifer Aniston's photos. And then there was a big buzz about it. Do we have a neuron for every identity we know? And what happens if we suddenly, we have a beer and this neuron is gone? Then are we left with no understanding of or recognition of Jennifer Aniston? And so a few years forward, we realized that a single neuron is in fact not so reliable. So the information cannot be residing within a single neuron, because if we now take a neuron and show a stimulus at time 0, okay, and you see this train of action potentials, these electric pulses, okay, and if you show this stimulus over and over and again, you could see that the neuron is very variable. The response is not very robust, in the sense that every time you show a particular stimulus, you will not see the same response, okay? And then once we understood that neurons are not so reliable, we got to the conclusion that the information is actually at the population level. So we need a lot of neurons because they are so noisy, okay? So we need to gather information from many, many agents in order to get an understanding of what the information we want to convey. Okay, so the information resides within the population level. Now comes the question, a technical question, and perhaps a very great challenge in neuroscience is that how can we, like if you want to record from a single neuron, it's not a problem. You stick an electrode, you record from a single neuron, that's it. But if you want to record from many neurons simultaneously, then it becomes really hard. If you want to do it in humans, it's almost impossible unless we remove the scan, right? And if you want to do it in animals, then you can't really have so many electrodes. You can have, there's a limit to the number of electrodes you could put inside the head. Okay, so if we look at the history of simultaneously recorded neurons, okay, so if we started in the 60s, we were recording from one, perhaps a pair of neurons. And then we started slowly ramping up the number of neurons that were recorded simultaneously over time. So it turns out that we have a doubling effect, a doubling effect of every 7 years, okay? So if we were to do the calculation, a doubling effect every 7 years means that had we wanted to record from the entire population of neurons in the human brains, it's going to take us 220 years to record from the entire population of the human brain. That's clearly not something that we are willing to accept, right? So what's our alternatives then? The alternative is looking at different, not humans, right? I mean, we could record many more neurons, but with the technology at the moment, we can record a few thousands, a few hundreds of thousands, something like that. So today, I'm going to show you some interesting results we got from very cutting-edge technology that is actually standing somewhere here on the graph, like very nearly about 1 million neurons, 100,000 of neurons. Okay, so basically what I am interested in is what we call this neural code. Okay, so the neural code has two ways to look at it. We can either show the brain or the animal as a visual cue, and let's assume that this is a population of neurons. Here it's denoted with 2 neurons, but it can be many more, right? And let's and this shades of gray is the strength of the response. Okay, so I show this dog once to the neurons, I get this response. I show it again, I get this response. I show it again, I get this response. So overall, one can look at the encoding model. Okay, basically, how does the brain represent this response given this stimulus? Okay, and we get this encoding model. And another thing that the brain does is, given a particular pattern, the brain asks itself, okay, I have this pattern. What is the likelihood of a particular stimulus given this, this response? Okay, so this is the encoding model, and that's the decoding model. And that's basically what we do. We try to understand how the brain represents the world and how this information can be then decoded from these active neurons. Okay, and what we will see today is how this code is actually developing, and something to do with the fact that the brain is also active when nothing is really being shown to the animal. So in order to address these questions, especially if you want to look at something over development and to look at the neurons at the very detailed level, one has to go to an animal. And today I'm going to show you this friend. So the animal model I'm working on is the zebrafish. Now you might be asking, okay, this is a fish, but where are the zebra lines here? And so I'll say that this is a mutant of the zebrafish that is lacking any pigment cells on the skin, which means that the fish is actually transparent. And because it is transparent, we can image the brain through the skin without doing anything invasive. So we can look at the brain as the animal behaves without doing any surgical intervention. Okay, so if we look at this fish, now this fish is very, very small. It looks like when I say fish, people think about a fish, but this fish is actually 4 or 5 millimeters long and 0.5 millimeter width, so it's super tiny. And if we zoom in onto its head, we see very big 2 eyes because this fish is very visually guided. Most of the behaviors that this fish is eliciting is driven by vision. And we are using some genetic tools that basically are labeling the entire population of neurons with some kind of a marker. Okay, so we have a very good ability to look at the neurons and to visualize them as the fish is sitting under the microscope. Now, if we are using very cutting-edge technology to look at this neuron at very fine resolution, we can actually see single neurons over the entire plane of the brain. And in addition, we can have a trip going into the deep, like we are we are imaging through the volume of the brain, and we are looking at all of the neurons in the fish brain. Okay, so in this fish brain, there's about 100,000 neurons. Okay, and we are able to record all of them. So this is the only animal at the moment that we are able to record the neural activity from its entire brain. So it's like a network that is complete and intact. We are doing nothing to the animal. We are not doing anything invasive. And that's the beauty of the technique. Okay. Now, once we have these neurons that are labeled, we can actually look at the neurons as they are in action. I don't know if you can see it with all of the slides, but basically what you should see is some bursts of brightness. So basically, whenever the neuron is firing, there's a change in brightness of the neurons. That this is how we know that the neuron is actually firing. Okay. So basically, what we do is we take this disco party that we record from the brain and try to make sense out of it. So here what you see is like neural activity of a fish that is seated in the dark. So we are looking at the visual areas of the brain, and you can see that the fish is still, the brain is still active despite the fact that the animal is sitting in the dark. It sees nothing, and still its visual brain is very, very active. Okay, so one can ask, okay, so you can record neural activity from the entire brain, and it's really fantastic. But what can this animal do? Okay, so I would say that this animal, although very small and very young, can solve one of the questions that we as humanity still not able to answer or to address properly. And that's, I think, perhaps now more than ever, the world is really interested in finding the question, and it is the question of shooting a moving target, right? So, and when I say shooting a moving target, I mean, usually when like the problem of shooting a particular target that has a ballistic or has some predicted trajectory, that's a solved question. But what happens when the target has some statistics that is unknown, unpredicted, and how can you then shoot a target that is moving with its own statistics, right? So it sounds like very funny that the fish is actually has solved that for its survival. If we look at the fish, now this is a dish. The dish is about 2 centimeters, and the tiny targets on the dish are living creatures. They are single-cell animals, paramecia they are called, and we are raising them in the lab. And basically what we do, we put the fish in a dish and put these targets, and now we are learning how does the fish approaches the target? What is the strategy to reach the target? Does the animal improve as it gets to the target? The fish, they are moving when they are young, they are moving in discrete bouts. Okay, so this discrete bouts makes it very easy to pass the behavior then, because we have a few decision points, okay? And then we can calculate some strategies, policies, okay? What's the policy of the animal once the target is here, it's there, and things like that. And the fact now this movie is slowed down in speed for your eyes. The fish is doing it in less than a second, okay? So it's very, very fast. Okay, so how do we try to understand what the fish is doing there from the behavioral point of view and from the neural point of view? So basically what we, the way that we look at it is we say, okay, there's a dynamic to the world, okay? The world is moving or transitioning between time points, okay? And also the brain is transitioning between time points. So there's the internal dynamics of the brain, and there's the external dynamics of the world. And basically the fish or us, we are experiencing the world by observing the world, okay? So there's an observation path which changes our brain activity. And then once we have this brain activity, we are acting upon the world, which again changes the state of the world, and so on. So it's like 2 Markovian chains that are interacting basically. Okay, so today what I'm going to show you is like a few like a glimpse into the way that we tackle these different questions. We will look at the quality of the code. How does the fish observe the world? Like what is this blue circles here? And then we will talk a little bit about the gray circles here, what happens in the dynamics of the brain. And then I'll show you some model, or at least the beginning of a model that tries to understand something about the actions that the animal is eliciting. Okay, so in order to understand this observation part, what we did is we placed, as I showed you, we placed the fish in a dish with a lot of targets, and we recorded from the top. This is 500 frames per second recording. It's a really massive data set because every recording is about 350 gigs of data, and we have hundreds of these recordings. And then once the fish has finished feeding, we are embedding them in another dish, and we are projecting. We have a small projector, everything is miniature, okay? So we have a projector. The projector is projecting something that is mimicking the prey, okay, a synthetic prey. And while the fish is watching this movie that we are projecting, we are recording the activity from the brain. Okay, and for this case, we recorded 30 minutes of neural activity in the dark. And then we showed this stimuli. We had 9 different stimuli. So every time there was like an on stimulus, and then we waited until the response of the brain decayed, and then another stimulus, and so on. And so we first connectivity. That's a very big question. We don't know. We don't have the blueprint of the brain. That's one of the biggest problems. Yes, you could. But so basically, there's a big project trying to dissolve the connectome of the fish. For humans, it's almost impossible, but for the fish, at least for very small tissues, you can. And Google is actually involved in trying to trace back all of the connections to have like a connectivity map. Yes. So when you do that and you try to look back, like the people do that, and whenever that happens, is like when you go back and see what the biology has to offer and compare it, it doesn't match. Yeah. Okay, so we first asked whether these fish are actually like they are born with this ability. Perhaps they don't even improve, and then there's nothing to learn about learning about development, right? So I first asked, do they improve with their hunting performance? So if we looked at them at different days post-fertilization. Okay, so that's how you age them. So we looked at them at day 5 and at day 15. And we looked at the durations of these events, of these attacks. And what we saw was that the duration of the attacks, they got shorter as the animal develops. Okay, so they become more efficient at executing this behavior. And then we asked how many decision points, how many actions did they have to elicit in order to get the target. And also for that, we saw that there's a reduction in the number of actions the animal was doing. We also looked at accuracy rates. Okay, so we looked at first at the ratio of events that the fish started hunting and then aborted the event for some reasons that we don't know what they are, and the ratio of the events that the fish has succeeded capturing the target. You can see that as the animal develops, it succeeds in hunting the targets much better. Okay, so behavior does improve. Now comes the question, how does the brain mediate these changes in behavior? And for that, I'll just say that when, as all vertebrates basically, when there is the visual, our visual world is mapped on our brain topographically, so that if there's 2 points in the world that are very close by, they will be represented in 2 different points on the brain that are very close by. So that's called a topographic map. And so basically what we did is we showed the stimuli at different positions in the visual field and collected responses to these different stimuli. Here you can see all of the neurons that were recorded, and whenever there is like a yellow patch, it means that the neurons elevated their firing rate. Okay, so they increased their firing rate. So for saying for a single neuron, one can, because we are showing it over and over again, for a single neuron, one can record the tuning curve. Okay, so that's the how well or what's the preferred position for this particular neuron. Okay, so we can see that for 45 degrees, this neuron didn't really respond so much, but for say 90 degrees, this neuron responded very strongly. Okay, so every neuron has its tuning curve or its preferred position. And then there's a notion that if there is this tuning curve, okay, every neuron has the area or the position in the visual field to which it is responsive. Perhaps when they grow old, these curves are getting narrower or become sharper, and that's what gives us this great acuity. But when we looked at the standard deviation of these curves, we saw that they didn't really change in width. What did change was the preferred position. Okay, so here I'm showing a cumulative distribution of the tuning for fish at day 4 and fish at day 15 and 5 and 9 and so on. And what you can see is that for young fish, most of the neurons are tuned for things that are occurring at the back. And as the neuron grows old towards the green curve, you can see that the visual field is represented evenly on the brain, as if the fish is born with some tendency to look backwards. And it makes a lot of sense, at least at the beginning, because the beginning, when fish are hatched, they are not feeding. For 5 days, they are not feeding. They have this yolk sac, and they are getting their nutrients via the yolk sac. So most of their worries is that someone will come behind them and catch them. Okay, we also looked at some measures of neural activity, the strength of the response and the density of the response, and we saw that more neurons are tuned, or more neurons were active in response to posterior stimuli at young age, and as the animal developed, this also evened out. Okay, so basically what we are looking at is some changes in neural activity as the animal develops. Okay, and comes the question, okay, of all of these questions, all of these changes that occur in the brain, can we decode better the information as the animal develops? And so we developed like a very simple classifier that takes the activity of all the neurons and try to estimate or predict what was the stimulus presented, in what position was the stimulus on. Okay, and what I'm showing here, that if we take the neural activity from the fish at different ages, we can see that the performance of decoding, we can actually decode the information much better when the animal is older than when it is younger. So the fish does improve in decoding, at least the information. In terms of information content, there's more information in the response of the neurons as the animal develops. And when we looked what exactly changes, okay, so you can see this is the decoding performance, but now as a function of the position on the visual field. And you can see that in the posterior areas, the fish can decode very well also to things that are coming from the back. But what does improve is the decoding of things that are occurring in front of the fish. Okay, so basically, so far I've shown that this animal does improve over developing in hunting. Okay, they become better hunters, they become more efficient, they become more accurate. And we saw some changes in neural activity, and we see that this neural activity not only changes, but we see that there's more information content. But now comes the question, can this change in information content or change in decoding performance be predictive of how well the animal will behave? Okay? Like, let's say I'm looking at a brain, I know what the decoding performance, can I say this is a good hunter or a bad hunter? And the answer was yes, we can do that. And so what we did here is we looked at fish at 5 days post-fertilization when they are 5 days old, and we saw that there's a huge variance between fish. Some of them are really good decoders, and some of them are really bad decoders. And so we asked, what's the relation between their decoding performance and their hunting performance? And that's what I'm showing here. So basically, the better decoder you are, the more information you have in your brain, the better performance, at least in terms of behavior, you will have. Okay, so this was the observation part. Now I want to talk a little bit about the what happens in the brain when nothing is really on, like no visual cues, and we are looking at the visual brain. Okay, so this is a visual brain in the developing mouse. And as you can see, what's very typical of it is that it's very synchronous. The activity is very synchronous. And that's why people tend to think that it's low dimensional and perhaps doesn't hold a lot of information. Whereas if you look at neural activity in the mature animal, it has it's of higher dimension, and it suggests that it might be holding some information or some more information relative to the young animal. Another thing that people found was that if you look at this evoked neural activity that is driven by sensory cues, and neural activity that is not driven by cues, but just by the spontaneous, something that is endogenous inside the brain, people found some similarities. And this raised some notions that perhaps this neural activity that occurs when there is no visual cues is acting like a Bayesian prior. Perhaps the animal has this spontaneous activity without any visual cues, and that's just expecting something to come. Okay, and people even reiterated it by saying that if that is the case, then if we take the similarity between spontaneous and evoked activity, this should be going down as the animal dissimilarity. If you take similarity, it will go up. So the two, because it's a Bayesian prior, then if you are trained on the world, you expect your prior to be very similar to your posterior at the end, right? So if this is the case, then the dissimilarity between the prior and the posterior will be decreasing. And that's this work was done a few decades ago, and a decade ago, and it was done using mainly a few neurons. And I suggested we do it again now with looking at the entire set of neurons to see what's the dynamics of this spontaneous activity in the brain. So again, we recorded this neural activity in the DAG, and then neural activity in response to this stimuli, and started looking at the relation of, or try to characterize the activity in the two modes, the mode with visual cues and the mode without visual cues. So if we look at the correlation structure of the population, in the case of the sensory-driven activity and the spontaneous activity, we see that at least in terms of correlation structure, it looks similar. So that suggests perhaps that there's this connectivity map that is driving this correlation. But when we looked at the relation of this, or the correlation between the correlation maps, we didn't find any increase in correlation. Suggests that they are not really getting closer to each other. We also looked at, we used some graph theory techniques to find these reoccurring patterns, okay, neurons that are reoccurring. So we built a graph of neurons, and where every edge was the correlation between the two neurons. And then we started looking for communities on this graph. And this is what I'm showing here. These are the communities, reoccurring communities on the graph. These were sensory-driven. The black ones are sensory-driven, and the gray ones are spontaneously generated. And then we asked, what is the variance? How similar the neurons in the evoked groups compared to the spontaneously driven groups. And you could see that the spontaneously driven groups were much varying in terms of their tuning. Okay, so this also gave us some hint that perhaps this prior or Bayesian prior idea is perhaps not really what is going on there. So we took another step further to try and understand what's going on there. And what we are looking here is 8 minutes during the activity in the dark, and then 8 minutes of activity, neural activity during the stimuli. And this is and what I'm showing on the y-axis is the number of neurons that were co-active. Okay, how many neurons were active at every time point. And so we looked at these time points where there's a peak in the coactivity level. Okay, and we characterized or classified a few different conditions. One were patterns that were occurring in the dark. One were patterns that were locked to the stimulus. And the third one were patterns that were not locked to the stimulus. They were within, between stimuli, but they were not locked to the stimuli. So this is to differentiate between patterns that are sensory driven and patterns that are spontaneously driven. And so we first looked at the similarity between these patterns. Okay, we have a vector, identity of neurons. This is a 1 and 0 vector saying these are the vectors that were active here, and these are the vectors that the neurons that were active here, and these are the neurons that were active spontaneously. When we asked how similar these vectors are, and what we saw was that as the animal developed, this similarity actually decreased between the vectors, like the representation of the world that were sensory driven and the spontaneous patterns that the circuit just delivered. And so then we looked at it from a geometrical point of view, trying to suggest that perhaps there's a plane, or perhaps, yes, a plane, a hyperplane that holds all of these patterns that were spontaneously generated. Here it's in a toy example, like a 2D plane that spans all of the patterns that were spontaneously generated. And then we looked at all of the patterns that were sensory driven and said, what is the relation between the patterns that were sensory driven and the patterns that were spontaneously generated? If they were a subset within this space, then the projection would be 0 because these patterns would be within this plane, right? And so we started playing with this idea, and the dogma in the field was that this is actually a subset, so that the spontaneous regime, that's including all of the patterns the circuit can generate, and the evoked patterns are the subgroup within this. But the results showed a different, the results told a different story. And the story was that as the animal develops, this pattern actually diverge. Okay, they become further apart from the plane of the spontaneous patterns. And this has led us to think about different geometries that one could explain the relation between spontaneous activity and evoked activity. And so basically what we now think is that there's a and again, this is a toy example and only 2D, but perhaps when the animal is young, these are the more transparent blue and red planes. Perhaps when the animal is young, these planes are closer, and as the animal develops, this plane develops some orthogonality, and this can serve the circuit to do some computations. Okay, because when you have something that is orthogonal to what you are being processed, right? So it doesn't interfere with your processing, but you can still include it and incorporate it when you are trying to generate like an adaptive behavior. It can hold some context, it can hold a lot of factors that the circuit needs to know while processing the information. And the last fun part I want to show is like a very cool trial of us trying to understand the red buttons here, the actions. Okay, so basically in the quest to address the question, why does the fish select this set of actions toward the target? Perhaps there's a better series of actions, right? What is the best policy? What's the optimal policy toward the action? And so we kind of played with the idea that perhaps we can define an action as a dynamical model, right? So there's this function f, And that's a function of the state of the animal and some control signal that pushes the animal forward. Okay, and how did we approach that? So basically what we did is we looked at the fish, we took the posture of the tail, and we generated a vector of angles with the horizon, trying to explain what the position of the tail is. Okay, so now we have a vector, and we have many frames like this. So we can use SVD and find these principal components that explain most of the variance in the tail flicks. Okay, and we found that with three principal components, we can explain 88% of the variance of the tail flicks. Okay, so now we have like an eigen fish, let's say, an eigen fish. And now I want to reconstruct the position of the tail given these three eigen tails. Okay, so basically, this is one posture, that's the black posture. You can see it here. And this posture is then being reconstructed using the first PC, the first and the second in the orange, and the three PCs in the yellow. And you can see that we are reconstructing it quite well. So if we look at now at the dynamics of these PCs, so in blue it's the real tail, and in orange is the reconstructed tail. And you can see, and this is the dynamics of the coefficients on the PCs. Okay, and you can see that this has a very interesting dynamics, and basically it's the entire behavior, it sits on a very interesting structure. Okay, so as you can see here, I'm showing it from one angle, and you can see this circle. But basically it folds. All of the movements of the fish can be described on a manifold. Okay, and now using this dynamic model, I can really explain with this model the actual movement on this manifold, right? So if I have here, this is a movement on X and Y, the movement that we saw just now here. So this circle, that's the movement. And now I'm using the model. So the model has a control signal that sends like a pulse. And then there's this dynamic that is propagating, and this is generating this movement. And when I put them both together, you can see that the model can fit quite well the behavior of the fish. Okay, so now instead of really explaining the movement of the fish with a lot of tail flicks, I have this delta function, a series of delta functions that can explain the entire behavior. So we saw this example, and now that's another example. This is another movement of the fish. Now we send a pulse, we generate this movement, and now you can see it one on top of the other, and you can see that the model can really capture the behavior of the fish. So the next thing to do is actually to see, okay, given now we can give a cost function for every delta function, okay? A cost for every delta function, and say, okay, what's the cost of this sequence? Why did the fish select this sequence and not another? Okay, so I'll conclude roughly saying that what we do really is we use maths and statistics to try to pass behavior and try to understand how the circuit is actually doing it. We are situated in this beautiful building with lots of neurons on top of it. It's very hard to see through the windows, but it's really fun and cool to do that. So thank you very much for listening.
Thank you very much. This is amazing. I have to add what I didn't say at the beginning about Lilach. When she started, she was against experimenting animals, and she wanted only to work on models for people. But now she's really Renaissance. She's really interdisciplinary. She does the mathematics, the statistics, the computer science from education, but she became a neurophysiologist, experimentalist, veterinarian, and electrical engineer because she's building also some. So thank you very much. You solved the question, how does the brain works?
Wow.
Any questions? Yes. I think you know it. Yes, please.
So the difference that you see from the young animal, is it like maybe plasticity with plasticity at the beginning, and it's rather straining plasticity once time passes learns. That's what the learning pattern.
Yeah, so plasticity is a big term. In the fish, it is known that by the age of roughly 7, there's not a lot of morphological changes anymore. So the increase in density of synapses and things like that, it kind of decays. And this suggests that it may not be plasticity. What can be is the addition of new neurons into the brain that can happen. And also, I think that every day, the experience of the fish has increased by a massive fraction, right? So I think that the fish has, every day, it adds a lot of statistics to its statistical repertoire, basically. And in a sense, we now have some other evidence showing that as they grow old, they are able to handle very like a more complex visual scenes because they know something about the statistics of the world. And if we change the statistics of the world, they become a bit like, okay, what's going on here? Yeah, so I think that every day that goes by, you could literally see the addition of the knowledge into the set. It's really beautiful. All right, thank you. Sorry, again? I have many of them, yes.
And work.
Yeah.
Co-activation. They
Right.
thought that change your organ, not the
Yeah.
activation.
So as opposed to really low animals that have, like, you know, in the worm, you have 300 neurons and every neuron has an identity, you know exactly where it is. And you call neuron 200, you know exactly where it should be. In the fish, and as you grow with the animal, it's very hard to know, like to say that this neuron is actually the equivalent in the other fish. What you can do is look at different structures in the brain. And this does is conserved across animals. So if you look at the patterns in different areas or areas in the brain, you would see that they are similar to other fish in the same area. Okay, in terms of compactness, in terms of the number of neurons within a pattern, things like that, all of the statistics of the patterns. But it's not that the pattern, there's nothing holy in the pattern, it's just a pattern. The thing is that this pattern holds some information. And these patterns, there's a lot of computational works that actually suggest that the pattern identity itself is meaningless because you can connect a stimulus with this pattern and comes tomorrow, you will connect it with a different pattern and you will still behave perfectly well. Okay, so it's not that the identity of the neurons matter. What matters is what they together say to downstream neurons.
Thank you very much.
Thank you.